T cells and cause them to re-infiltrate. And this is accomplished by decorating the cell surface with receptors that cause it to home to the tumor site. Or we can, in the case what we're going to discuss today, is to locally administer a cytokine, which then also will cause the cells to home to that site. So actually, this treatment in oncology is a complex interplay of cytokines, natural killer cells, which are basically the, using the army analogy, are those troops that actually do the work, and the T cells, which regulate and direct. So why did we choose IL-12? IL-12 has been studied in the literature. A number of years ago, they attempted a number of trials in the clinic with it. However, what they found when you give this intravenously, remember the example with the GPS, if you give the IL-12 intravenously, what happens? The cell's home to wherever the signal is, so you get the off-site recalculating type of thing on your GPS. You then slam the GPS against the wall, and you move on. So anyway, so IL-12 is a pro-inflammatory cytokine. It's a master regulator of cell immunity and pathogens, and leads to neoplastic transformation. It's also produced by the innate cells in response to pathogens. It leads to the production by T cells and natural killer cells of gamma interferon, as well as TNF alpha. So these, and that leads to the homing of different cell types and downregulation of T regulatory cells. So, like I said before, our technology, we have to in some way control this, because if it goes unopposed, we'll end up with destruction. So to accomplish this, what we developed in collaboration with Intrexon group is an adenovirus, which is replication incomplete, to which we have a switch that is attached to it, and then attached to that is an inducible gene cassette. So we can switch out the gene cassette, since it is making it a platform technology. So what happens is when we give an oral activator ligand called Valetomex, that causes a conformational change and then turns on the gene to produce, in this case, IL-12. So how does this work? Here is data to show that it does work in vitro. This is in HD 1080 cells, where we transduce them with the adenoviral vector to make IL-12. We then incubated with 75 nanomolar of the Valetomex. And what we see in the presence of the Valetomex, we see an increase in IL-12 protein production. We then wash, and we see that within 36 hours it's back to baseline. And then we can actually stimulate again, and it comes back up. This can be done 4 or 5 times in a row. So now that we know in vitro that it works, does it work in vivo? So this is a study that we did in 4T1 syngenic mice, which have an intact immune system. We placed tumor cells into the flank and allowed the tumor to grow to between 100 and 200 cubic millimeters, and then administered the vector intratumorally. There it is. As well as then administering the Valetomex orally. So the first step in this, obviously, is do we get the vector in the tumor at a consistent level? Yes, we use the same dose of vector for all these studies. So it shows that you can, in fact, inject the tumor and get the same amount of vector present. Administering the activator ligand Valetomex, we see a nice dose-related increase in tumor levels of Valetomex. So we have both components present at the site of the tumor. We then looked at the mRNA using RT-qPCR, and we see a nice dose-related increase in the mRNA. This then translates into the dose-related increase in the IL-12 protein produced. And we also have looked at the interferon gamma, which I don't have the slide for, where it also shows a nice dose-related increase in interferon gamma, demonstrating that the IL-12 we're making is in fact viable. So here is a immunohistochemistry staining comparing the vehicle with animals treated with the vector plus Voletimex. What we see is an increase in infiltrating CD8 T cells in the tumor, as well as an increase in the CD4 helper cells. Looking specifically at the T regulatory cells, which are FOXP3, we see that there is a marked decrease in those cells. So we know we're on mechanism. Does the stuff in fact work? That's the important thing. And here we did a study in the same 4T1 xenograft model. And what we see here are your controls in green. And we see a nice dose-related shift in tumor growth rate, indicating that the compound is on mechanism and does in fact reduce tumor mass. So with that in hand, we went to the FDA and opened an IND, and we enrolled 12 subjects in a trial that was a 21-day cycle with 6 cycles of the vector Voletimex combination administered. And dose ranges range between 80 and 140 milligrams flat dose, and the Voletimex was administered once daily for 7 consecutive days. So here's the demographics. What's important to note is that we have a large number of subjects that are stage 4 breast cancer. Also, they have 92% had metastatic disease, and the average number of prior therapies was 13. So these were heavily pretreated individuals, as you would anticipate in a phase 1 trial in oncology. So what we observed is that, like in the mouse, we observed that we could, when we add the Voletimex plus the vector, we see an increase in IL-12 and an increase in downstream interferon gamma, indicating again that we're on mechanism and the IL-12 that we're producing is functional. So here we have waterfall plots of the response to these individuals. So the important thing here is per RECIST criteria. If you manipulate the lesion, you can't count it in your analysis. So we are physically injecting the lesion, so we cannot count that. So what this is really looking at is this is looking at a therapeutic effect in other lesions outside of the primary lesion. So essentially a therapeutic vaccine. Yeah. And what we observed here on the left is the best overall response on a per lesion basis. And we see that the average number of lesions the subject had was between 1 and 4. We see in 2 different subjects, we see very good PRs. When we look at total tumor burden, we had 1 PR, and as luck would have it, they developed a new lesion on cycle 6, day 7, which is the last day of dosing. However, they are still doing fine, and they have not succumbed to the disease. And this is over a year out. So with this in hand, we began to do some work in glioma. I'm the non-clinical guy, so I've already done it in breast. What else can I do? So I moved into the brain. Since the brain is an immunoprivileged compartment, I thought that we could lower the doses a lot lower and get the infiltration of the T cells into the brain and have a dramatic effect. So in this particular model, this is a GL261 orthotopic mouse model. What we do is we drill a hole in the brain, 1.5 millimeters lateral to bregma and 1.5 millimeters anterior to the bregma. Administer 1 times 10 to the 5th GL261 cells. We then seal the hole with a little bit of bone wax. Come back 5 days later when the tumor has started to grow and administer a single dose of the adenoviral vector. We then follow up with Voletimex in this case for 14 days. And in the studies I'm going to show you, we compare it to the traditional standards of care. And we look at the time to progression and/or survival. So the first question is, does the Voletimex cross the blood-brain barrier? Because obviously if it doesn't, we don't have a drug. And what we observed here, this is in normal mice. We see in normal mice we have a nice dose-related increase in Voletimex in the brain. And then in the GL261 tumor mice, at the beginning and at the end of dosing, we also see increases in Voletimex concentration. Importantly, we range between 1.7 and about 10 times the amount of valetomex in the tumors in the brain of animals that actually have a tumor versus those that are normal. So again, now that we know that, we administered the vector and the valetomex together into the valetomex is administered orally. And what we observed, like we observed in the 4T1 mouse, we see an increase in the messenger RNA, and we see an increase in the tumor IL-12 protein, as well as the downstream interferon gamma. So here is your Wall Street money picture that every vulture capitalist likes to see. And what we see here, this is a normal mouse brain. This is the normal progression of the disease in the mouse by day 20. You can see that this particular mouse has a huge tumor, and actually the animal, we had to euthanize it. So 74 days after the institution of therapy with the valetomex plus the vector, we see that we have the same brain. So this animal is in fact tumor-free, and it's confirmed by pathology. So here is the Kaplan-Meier survival curves. So as I alluded to before, and you saw in the previous picture, the vehicle alone, they also come by day 20. The vector plus saline, for whatever reason here, we had 2 mice that did not die. When we administer valetomex plus the vector, we see a nice dose-related increase in survival, with survival of greater than 60% over 80 days. The Iacocca would not let us go further than that, and those of you who have dealt with Iacocca, you know it's an uphill battle. To compare to the standards of care, we looked at bevacizumab, temozolomide, and an anti-PD-1 inhibitor. What we observe here is that the valetomex at IL-12 combination is superior to the standards of care. With that, I'd like to point out, though, that the standards of care do, in fact, work, because if we look at temozolomide here, and we come across and look at the 50% survival, we have doubled the 50% survival. So this, in this assay, this would be indicated as an active compound.
Yeah.
We did encounter some problems, however, when we wanted to do the same analysis on these animals, because surviving animals per Kaplan-Meier are censored. So that did not give you a very good indication of what was going on. So in the next study, we actually took these surviving animals and we re-challenged them with GL261 cells. And we compared that versus age-matched controls. So here we have the age-matched controls. Keep in mind these mice are now 3 months older, so they do not succumb to the diseases rapidly. Here we see that we have over 90% of those animals that were previously treated remained alive and tumor-free, indicating again that we have systemic immunity. So with that in hand, we went to the FDA again, and we started a phase 1 clinical trial. And this particular design is subjects that was scheduled for a 2nd or 3rd surgical resection. What we did here is we administered the valetomex about 3 hours before the tumor was excised, so that way we could get the tumor levels of valetomex to demonstrate that, in fact, it crosses the blood-brain barrier in humans. And then, like the mouse, we dosed with the valetomex for 14 consecutive days. Demographics of this population, it's a tough disease. The median age is 40, and basically they had all between grade 3 and grade 4 glioma at entry. Looking at the side effect profile, the side effect profile is consistent with the mechanism of action of the compound. We did see some aseptic meningitis. We saw neutropenia and leukopenia. However, these are not the classical neutropenias and leukopenias. This is more margination than it was in fact the way you get from frank bone marrow suppression. That removal of valetomex within 48 hours, the values came back to normal. So again, looking, does it in fact cross the blood-brain barrier, get into the tumor? We see, like the mouse, we see that it does in fact get into the tumor, and the ratio is slightly higher than that we observed in the mouse. About 25% of the administered dose is present in the tumor. So do we produce cytokines? So here we have IL-12 in the brain, in the serum. Keep in mind, in the serum, the values are about 15 to 20 times less than those in the tumor based on mouse data. And what we observed at this very low dose of 10 milligrams per meter squared, keep in mind we were at 450 in the mouse to show efficacy, we see that we have IL-12 production. And this one particular subject here is interesting. Being a pharmacokineticist, the study site put them on a CYP3A4 substrate, and this is a CYP3A4 substrate also. And what we observed is, as you would have anticipated, a much higher blood level. Likewise, we do produce downstream interferon gamma, indicating that it is biologically active. We also looked at the inhibitory cytokines, IL-10, which is upregulated in response to IL-12, and IP-10, which is upregulated in response to interferon gamma. And as you would anticipate, we see increases in both, mirroring the increases we see in the IL-12 and the interferon gamma. So in summary, and this was a very fast, rapid talk, I realize, that the combination of the viral vector plus Voletimex elicits controllable systemic immune activation in human subjects with breast and glioma. We also observed a dose-related increase in plasma and brain tissue with no accumulation in the brain. That was from the mouse data. The vector plus Voletimex improves survival over the current standards of care, as we observed in the mouse. And those 5 subjects are all still alive and doing okay. The first subject is about 225 days out. Normal disease progression time is about 3 months with the second resection. And obviously, we feel that these findings support the utility of this approach in the treatment of glioma and breast cancer. So with that, I'll stop and entertain any questions.
I think you know this is one of the toughest tumors to deal with. Yep. While it doesn't metastasize, it just grows right. Indefinitely locally. No, and so that when it's debulked surgically, you still have the arms of it continuing to grow. Now, Temodar was not in this, right?
Yeah, it was temozolomide.
Or it was in this. Yeah. Okay. So there was another drug on board. The blood-brain barrier is something we worry about all the time. In the animal models, did any get across the blood-brain barrier in normal brain that was not involved?
Yeah, I showed that figure actually, and
I may have been out of the room.
Yeah, the mice. Well, actually, we have a little bit of time.
That's all right.
Almost there. Oh, almost. There. So there we have on the left here. This is the normal mouse at 2 doses. And
good.
Then
Okay.
This is the tumor mouse.
Terrific.
So showing it across the blood-brain barrier.
Terrific. Thank you very much. Questions here for Dr. Barrett?
Have you ever noticed NK cells or any lymphocytes during the process of tumor clearance or the anti-tumor effects?
Yes, we have significant numbers of NK cells present. I truncated the talk because I thought I only had 20 minutes. I have all of the fax panels and things like that to go along.
So on that note, does it mean then IL-12 regulates the receptors for the NK cell ligands to bind, or is there any change in the regulation or the expression of the receptors or ligands that are compatible for the NK cell detection?
So that we're looking into. We know that we see an increase in NK cells. Whether there's an increase in the dendritic cells that present it to the lymph node, at this point we're still investigating that. So, the immune system is very complex.
Yes.
When you look at any of the review articles on the checkpoint inhibitors, people forget you're either looking at the antigen-presenting cell and the white cell and forgetting the tumor cell, or you're looking at the white cell and the tumor. They forget that there's a third piece to this.
Yes.
What is the chemical nature of Veledimex? Is it chemical synthetic molecule or nature-derived molecule?
Yeah, Veledimex is a phenylhydrazone derivative. It's a small molecule, and it was isolated from insects' endosome. So it's not something naturally occurring in humans, so we would not see the switch turn on in humans.
Yes. Hello.
Yes, great presentation and very nice work. A colleague of mine, Nejat Egelmas, he did a lot of work with IL-12. Probably you're familiar with his studies, but he's shown that he was using PLG delivery system or injecting IL-2. Initially, you get a pretty robust response against the tumor, but later on, tumor becomes resistant to IL-2 treatment and comes back. And if I recall correctly, what they found out is that in those tumors, you have T regulatory cells that are expressing fast ligand. They eliminate CD8 cell through that mechanism. And it appears that you don't see this in your system, or are you familiar with this, or any explanation? Is it because the way you design your system is not necessarily, it's a controlled delivery type of thing? I'm just wondering.
Well, yes and no, unfortunately. So the adenoviral vector, the gene is controlled by the activator ligand. So we can throttle up and throttle down the amount of IL-12 we generate. What we did observe in both the mice and some of the human studies in the breast cancer was that it was in fact a decrease in the T regulatory cells. We have not looked at the FAS and the FAS ligand yet.
You also got pretty high level of IL-10, other immunoregulatory molecules. And I don't know whether you have any explanation as to what IL-2 is doing. Is it positively impacting the immune system, or is it negatively? Because it's a controversial cytokines.
Yeah, I know. No, that I can't answer. I don't know, actually. So.
Thank you.
Any other questions? Some other studies, for example, with herpes simplex virus as a carrier, have run into trouble with insufficient reaching of all the cells.
Right.
I mean, it looks like you really have access to all the cells here. It's really very exciting.
Well, the herpes simplex, there's different kinds of vectors. So there are replicative complete vectors, which the herpes simplex is. So basically, you're injecting herpes into the tumor and hoping that it self-eradicates.
Yeah.
And likewise, Duke, I don't know if you've seen that on 60 Minutes, where what they did, the reason it took them 7 years to get into man was they were injecting the active polio virus into your brain tumor. So this vector is just used to transduce the cells. It is not replicative. So that's a
Very
real
important difference, to say the least. Good. Thank you very much, John. That was excellent.